Hello, everybody. Thanks for being here for this Friday talk. Today we have Andrea Bravo and the Mercurius. So I didn't know that, but it's going to be a choral presentation, I think. Andrea, well, she did the PhD in Switzerland in 2010, and then she spent many years as a postdoc in Sweden until she came back to Barcelona, and she has played all the roles of the AUCA, which means Beatriz de Pinós, Marie Curie, Ramon y Cajal, until she got a position as an investigator here at the center. Her work is about mercury in the ocean, the biochemistry of the ocean, especially the toxic methylmercury. And I think today she's going to talk a lot about what she's doing with her team. So, Andrea, thanks for accepting to make this.
Thanks, Ramon, for the invitation. Thanks a lot for coming. When I actually accepted the talk and gave the title to Ramon, I thought, okay, progress and challenges in understanding methylmercury formation in the ocean. Everyone from the research group I'm leading could give this talk, actually. So then I didn't want to give it alone. We are going to lead you, or we are going to guide you through the mercury cycle. All together, each of us is going to present more or less the parts they are focusing and improving knowledge. So, why mercury? It's harder and harder actually now that we don't have thermometers with mercury to show that mercury can be a liquid metal. And before, all of the thermometers were like this, and once a thermometer was broken, what we could do is to play with these little balls. That actually is not recommended, but most of us, we have done it. The problem is that it is extremely toxic in very high concentrations. The first case where first time we realized how toxic was mercury, it was in the Minamata Bay. And actually, one company was releasing mercury to the bay. People were consuming the fish of the bay, and during more than 20 years, they were intoxicated in mercury. I put often this picture. It's not a very nice picture, but actually it shows very well how dramatic can be mercury poisoning and intoxication. And the photograph, Eugene, received an award for this. That happened in 1956. It was a long-term mercury exposure that we actually recorded. A few years later, in Iraq, actually, we observed a huge case of mercury intoxication. In this case, it was a short-time exposure. This is quite important, because mercury has been used as a fungicide for a long time. And what happened is that the delivery of the seeds arrived late. So they didn't have the time to plant the seeds, protect the seeds against fungi. They used the seeds directly to produce bread and different products for direct consumption. And it caused, recorded, between 500 and 2,000 deaths in less than 2 years. That was in the 70s. So then the next, 56, 22, what about nowadays? Come on, Andrea, you are giving us this talk, and that was very long time ago, right? Well, still many of the products that we are using, most of the phones that we have in our pockets contain mercury because it has really nice properties and it's it keeps very well the electricity and so on. Okay, let's go on the news. Just Googling. So still in 22, we have cases of mercury poisoning. 23, it has been related, high concentrations of mercury have been related to rates of suicides. And a lot of people, actually, and many other news, you can Google, relate mercury poisoning with the consumption of fish. The famous tuna cans, actually. From time to time, we can see in the news that they have found tuna cans with high mercury concentrations. Indeed, we have a case of extremely exposure to mercury among our society, so it's not that far. It's not US, it's not Japan, it's not Iraq. It can be actually here. And actually, Jorge Fernandez explained much better than me, because he was intoxicated, how bad can be mercury poisoning. So, you are going to listen to him. If I manage all these technological things.
Mucha suerte, sabe que estás cuadrado y también saben que lo pasaste mal con varias enfermedades. ¿Qué ocurrió?
Pues que yo empecé a sentir mal hace como 7, 8 años, me empecé a sentir mal y no sabía por qué. Y yo me cuido de la alimentación, hago deporte, gestiono mi sueño, o sea, todo lo que parece que hay que hacerlo bien, yo lo hacía y me empecé a sentir muy mal. Y resulta que por un lado tenía un síndrome de Lyme, que es la picadura de una garrapata. Vi que tenía mercurio también, muchos niveles de mercurio, porque yo durante 15 años de mi vida comí muchísimo atún, muchísimo, pero muchísimo es muchísimo.
Todos los días rollo.
Pues rollo que me hacía los sándwiches de atún para ir al gimnasio, porque el atún es proteína, omega 3, y yo no sabía que el atún tenía mercurio. Entonces yo me ponía morado. Entre una cosa y otra, pues yo no sé si habría un día que no comiera atún. Así durante 15 años de mi vida, pues yo tenía unos niveles de mercurio brutales. Que el mercurio lo que es disruptor endocrino, es un desestabilizador hormonal, y eso te fastidia todo el organismo. Todas las hormonas están como bailando, ¿no? Bueno, pues yo me hacía analíticas y joder, pues la TSH por arriba, la testosterona por abajo. Yo adelgacé 12 kilos y no sabía por qué.
Okay,
Mucha gente de España sabe que eres el presentador de la Ruta de Suerte, sabe que estás cuadrado y también.
Of a whole interview that he made, and you can find it in YouTube. It lasts for 1 hour something, and actually he explains all the symptoms he encountered because he was intoxicated by mercury. Indeed, it has been reported that exposure to methylmercury, still nowadays, it means economical losses to the different countries, and it can reach actually 100 billions. Because of this, in 2013, Spain signed the Minamata Convention, which is legally binding and aims to reduce mercury emission, decrease mercury levels in the environment, and identify sources and processes. In 2020, in the Monaco Commission on Human Health and Ocean Pollution determined mercury as one of the top issues of ocean pollution. And actually, at the CSIC, in the White Book of Ocean Health, mercury was identified as one of the main problems in the ocean. So, okay, how this mercury reaches and arrives to us? So, mercury can be emitted by anthropogenic sources and by natural sources. When in the atmosphere, it can travel very long distances, because if you look at the screen, you will see one chemical species that is Hg0, which is extremely volatile and can travel 1,000 kilometers and reach the ocean. Once in the ocean, it can be, so how it can reach the ocean? It can reach the ocean by atmospheric deposition, as you can see here, or of course, by river discharge. Okay, once in the ocean, it can be methylated to form this chemical form, methylmercury, which is the chemical form that is bioaccumulated into the food chains, reach this famous tuna, and arrive to us. Methylmercury can be degraded, and all these chemical form reactions can happen all the time. So mercury can be oxidized, methylated, demethylated, and reduced again. And most of these reactions are performed by microorganisms, microorganisms that are carrying specific genes. So this gene is responsible for the formation of methylmercury, this gene is responsible for the degradation, this gene for the reduction and potential elevation. Okay, when we look at the budget of mercury in the ocean, and we look at the last UNEP report on the global mercury assessment, we realize that we have these two main sources to the ocean, as I have described, right? Deposition and rivers. But I was super lucky to actually work with Clara and have with Clara, and she made me realize that actually submarine groundwater discharge could be actually an important source, at least a characterized source of mercury to the ocean and to the coastal areas. And related to this, we have two postdocs that are actually responsible for really digging into the role of these coastal aquifers. And Celine, who could not attend it, unfortunately, today, is gonna tell us about it, if I manage all the technological challenges. Ahora no. It's gonna come. Okay.
The coastal aquifers are connected to the ocean by submarine groundwater discharge, or also called SGD. They have been described as a source of nutrient, for example, to the coastal ocean, but they have been clearly overlooked as a potential source of mercury or methylmercury. We know that the concentration of methylmercury or mercury in groundwater may change a lot according to the characteristic of the aquifers, among others. But our research has shown that coastal groundwater in the Spanish Mediterranean coast are generally enriched in total mercury, and that SGD is a source of total mercury to the coastal ocean. In particular, in the Mar Menor coastal lagoon, the recirculation of seawater that can be stored into the aquifer from weeks to years is actually the main process that releases mercury to the lagoon through SGD. And in this site, the flux of mercury through SGD is similar compared to the known atmospheric deposition. Now the question remains about the formation of methylmercury in these underground systems. Our first results suggest that when the groundwater are rich in nutrient and poor in oxygen, they may favor methylmercury formation. And this is supported by the presence of the methylating bacteria called geobacter. So in this condition, methylmercury can be formed in the aquifer and along the shore in the sediment, for example. And it can be released through the SGD to the coastal ocean, where it can be transformed or deposited.
Okay, so actually Celine and also Gemma working on this, as they have said, they actually unveiled that actually these SGD sources can be as important as atmospheric deposition in some of the areas. So by still looking at the potential sources of mercury to the ocean, we look at the previous, so at the UNEP report, and we realize something that it was a clear mismatch. And it's the actually geogenic emissions. I don't know if you see the number, but here is written 100. Geogenic are volcanoes, for example. And then when we look at the land, we see that this number is 500. But we all know that most of the volcanic activity is happening underwater. So actually we wonder how much the geogenic sources that are underwater may contribute to the mercury cycling and the processes. And this is going to be explained by Esther. Esther starts really fast. But
Hello everyone, I am Esther López, a predoctoral researcher at l'Université de Pau de Pays de la Dour in France and the Universitat Autònoma de Barcelona, l'Institut de Ciències del Mar, and I'm very interested in hydrothermal vent systems as the only source that directly emits natural mercury to the ocean. Volcanism has historically been associated to terrestrial environments, while up to 80% of Earth's volcanic activity occurs underwater, right where the biological methylation and demethylation processes take place, being the gateway for organic mercury into food webs. Although previous studies have confirmed hydrothermal vents as sources of mercury, these still remain poorly understood and likely underestimated. For that reason, we conducted an exhaustive research of the AGC genes responsible for mercury methylation in more than 1,000 metagenomes associated to marine hydrothermal vents, retrieved from public databases. And we found that putative mercury methylators were widespread in hydrothermal vent systems, with statistically significant differences in AGCA gene abundance across oceans, being the Indian Ocean the one with the highest abundance. In addition, putative mercury methylators were found in 4 different habitats, including sediment, biofilms, chimneys, and vent fluids. Well, in this study, hydrothermal vents were also classified within 6 different vent types according to their geophysicochemical characteristics. And what we found was that putative mercury methylators were present in all of them, ranging from sulfide-rich systems with temperatures up to 400 degrees Celsius and a pH as low as 2, to carbonate-dominant systems with temperatures around 40 degrees Celsius and highly alkaline conditions reaching pH 11. So, these results show us that mercury methylators are able to thrive in a wide spectrum of anaerobic conditions, and the hydrothermal vents are potential hotspots for mercury methylation.
and this is third PhD thesis, that actually really will focus and will try to unveil the role of these hydrothermal vents, especially surface hydrothermal vents that are more directly linked to where the fish that we consume are living, and the genes involved in the formation of methylmercury. Okay, we clean a bit the whiteboard. We know, okay, this is the mercury cycling, these are the chemical species, these are the different sources, but there are also outputs from the ocean. Not everything that is coming is there and being transformed. Actually, there is some sinking to the sediments, so the same reactions that happen in the water column can occur also in the sediments. And there is evasion. And actually, the evasion is controlled or can be controlled by the stability of the surface microlayer. Gemma Casas.
Hola. Okay. The reduction of ionic mercury 2 to volatile mercury 0 is a key process that controls the mercury exchanges from the ocean to the atmosphere. This begins in a... This begins in a really thin but crucial boundary that it's called the sea surface microlayer. The sea surface microlayer is the first micrometers of the sea surface, and it has really different properties, physicochemical and biological properties, compared to the underlying water. It's rich in organic matter, surfactants, and a specialized bacteria called bacterioneuston. With these unique properties, the SML, the sea surface microlayer, is the hottest spot for mercury exchanges and transformations. So every year the oceans emit 1,900 tons of mercury in the atmosphere. So this is more than a half of the mercury, the total mercury that it's released into the atmosphere. And so the ocean, this means the ocean acts both as a sink and a source of mercury to the atmosphere. And I also wanted to say that the sea surface microlayer, it's important to study because maybe we are creating a bias of the calculations of the mercury, of the total mercury globally in the fluxes from the ocean to the atmosphere. Despite the importance of the sea surface microlayer, is still poorly understood. And most of the research are focused in organic compounds or gases, and trace metals are rarely investigated, and for mercury there is only one published study. So, and for all these reasons, the aim of our study is to fill this gap of knowledge and to understand if the SML composition is important in the global fluxes of mercury. So during the PANTOC project in June 2025, in the campaign that I participated, we analyzed mercury 0, total mercury, and methylmercury in the sea surface microlayer, in the underlying water, and also we collect samples in different depths from the water column. And we are also studying how different physicochemical and biological properties can control the concentrations of mercury in the sea surface microlayer, such as organic matter, the wind, the wave, the surfactants, or the bacteria. So these are really new results. I hope I will explain you soon all the results. But so in these early results, we could see that the concentrations for most of the samples, the concentrations of total mercury were significantly higher in the sea surface microlayer than the underlying water, having an enrichment factor between 1 and 2. So this is really important, and it makes like a result, when it's a previous result, to say that the skin of the ocean, it's really important to understand the evasion of this mercury to the atmosphere, and also to control and to do good calculations of the mercury fluxes between the ocean and the atmosphere. And that's all. Thank you.
So, okay, we have right now focused more or less in the inputs and outputs and how the skin of the ocean can actually control the outputs. But let's focus actually in what are the processes that are occurring and that can lead actually to the formation of this chemical form that is bioaccumulative and to which we are exposed. So, as I have said, methylmercury is formed by the activity of the microorganisms that are carrying this gene cluster, AGCA. And I hope you are going to keep this name, AGCA, because it's going to be, from now onwards, a keyword. So, when the gene was discovered, so not so long ago, actually, 2013, we started to see in all the databases and cultures where this gene could be found, in which microorganism. And we realized actually that it was much more diverse than we thought. Previously we were thinking that mainly sulfate-reducing bacteria and iron-reducing bacteria were the methylators. Now that once we put the gene, we were able to expand the phylogenetic tree and see that they were actually more diverse. Key role of these functional groups all around the ecosystems, but we realized that also archaea and other microorganisms were responsible. In 2020, because the genetic tools in the last years have improved a lot and the databases have improved a lot. So for this case, more than 200,000 genomes and 1,000 of them contain the AGCA, improved indeed the description of mercury methylators all around. And just in 2020, many more were discovered, and we are still in this point. We are searching and discovering mercury methylators and the habitats where they live. But something that we don't know or we don't say often is that they are anaerobes. All mercury methylators that have been described with the potential to form methylmercury are anaerobes. So then we went to ecosystems that we know they have water columns oxygen depleted, such as the Black Sea. In the Black Sea, we didn't go, so that you know. They collected in 2018, Rosati et al. Went to do a study where they collected samples to study methylmercury concentration here in this plot. And they studied different water layers. The oxic layer, which is highly concentrated in oxygen, the suboxic layer in which oxygen depletion is happening, so we are losing oxygen in these layers, and this anoxic layer, where we don't have oxygen at all, and in the case of the Black Sea, we have the production of sulfides. And what we could see is that, of course, methylmercury was increasing while decreasing this oxygen and increasing this sulfide. What about the AGC? What about the microorganisms that are performing this process? They are also increasing. You can see here the abundance of different functional groups, in white firmicutes, in gray the sulfobacterota, in dark archaea, which of course appears when we have lost most of the oxygen. We then went to another basin where we know there are anoxic waters, the Black Sea. Here you can see, I will try to guide you. So this blue line is oxygen, okay, this dark black line is sulfide. So we went to one side where we know that oxygen was consumed very rapidly and at the same time almost sulfide was produced. And then we went to another side where this redox transition was bigger. So here oxygen is consumed quite fast, but the production of sulfide, so the activity of, for example, sulfate-reducing bacteria, does not appear until almost 100 meters deep. What about mercury? Okay, in both cases, actually, we observed that methylmercury, here in black, it doesn't see very well, but okay, it increased when the oxygen is consumed, and then it is maintained the concentration more or less up to the deeper points. And in the other side, actually, it started to be produced somewhere in the middle, somewhere in the middle on the redox line, so where this oxygen is consumed, and increasing with depth. Okay, can we relate this to the microorganisms? So then we, through metagenomics and metatranscriptomics, we reported the gene counts. Okay, when the oxygen is depleted, we have this increase on the AGC gene counts in blue, or even the transcripts also in gray. So maximum methylation and transcription occurs where we have the maximum methylmercury formation. And in this case, it was a particular case because it does not happen in all the ocean basins we have studied. We were able to actually modulate the concentration of methylmercury from the transcript abundance and the chemical speciation of mercury that is determined by the concentration of oxygen and the appearance of sulfides. But something that actually appealed to our attention when in the Baltic is that we observed actually in few of the samples that in the anoxic layer, of course, where we detect, here I guide you. So here we see the abundance of the genes in different layers. Hypoxic, anoxic, hypoxic, anoxic. And something that we, of course, thought in the anoxic layers, abundance will be higher. But something that we didn't know, we didn't actually look for at a first instance, it's in the particle size of the pore size that was bigger, so this filter retains the bigger particles and the bigger microorganisms in theory, we found a higher abundance of mercury, methylators. So we are trying to understand the role of particles as a hotspots for methylmercury formation in anoxic basins. And Julia is going to explain about it.
Okay, so one of our projects aims to figure out if and who methylates mercury in the Cariaco Basin, which is located off the north coast of Venezuela. The Cariaco Basin is one of the largest marine sulfidic systems that we have on Earth, and this means that it has hydrogen sulfide at the bottom. So in order to explore this, we use the Marquicoco pipeline, which is a bioinformatic tool that serves to detect AGCA genes in different metagenomic communities. And for this, we looked for these genes in both the particle attached and the free-living metagenomes from the water column of the Cariaco Basin. So here what we have is the depth in the y-axis and then the AGCA abundance on the x-axis. We show data from two different sampling dates, and we show the particle attached associated AGCA genes as like painted circles. No, sí, esto da igual. And the free-living ones like unfilled circles. So basically what we saw, so we did not detect AGCA genes in the oxic layers of the water column, but when oxygen started to drop, we began detecting these genes along the water column. The abundance peaked when the concentrations of hydrogen sulfide were the highest, and we also observed that all the time we got more AGCA genes related to the particle attached fraction compared to the free-living fraction. This graph here shows the depth, again in the y-axis, and then the contribution, the proportion of the different phyla contributing to the free-living communities and the particle attached communities. And also what we saw is that the sulfobacteriota-like AGCA genes dominated in the water column, both in the free-living and the particle attached communities. We also observed higher diversity of AGCA genes in the particulate fraction, maybe due to the fact that these particles offer a more beneficial niche for microbial communities to coexist. This could be one of the reasons why these particles are considered potential hotspots for mercury methylation.
Okay, so we have got into the processes, no, on the gene. And okay, we have described many of these anaerobic environments. And what is happening actually in the ocean? That anthropogenic global change, it is right now contributing to the expansion of oxygen deficient areas. In this map, what you can see actually is the loss of oxygen, so how much oxygen is being lost in different areas, and actually directly the red dots are hypoxic areas. So coastal areas are especially sensitive to this global change, and on top of this they are especially sensitive to the sea water levels rise that are encountering. And in this context, not so far from here, the Venice city is a especially vulnerable ecosystem.
Hi. So we use Venice Lagoon as a case study for these global change-induced alterations in the mercury metabolism. As you must have heard, flooding events have increased in the late years in Venice, causing millions of euros in damage caused and invaluable cultural losses. So in order to avoid these floods, the city has developed these mosse, which are barriers that can close the lagoon on demand. They usually close the lagoon for periods of 4 or 5 hours in autumn and winter, but sometimes they need to close it during summer and spring where temperatures are hotter, warmer. And they have even closed the lagoon for periods up to 2 days. So as you can imagine, the biogeochemistry of the lagoon is affected severely by this activation of the barrier. Why is that important in Venice specifically? Because for the first half of the 20th century, there was an industrial plant working with chloralkyl compounds that severely polluted the lagoon with mercury. As Andrea has been saying, low oxygen and mercury is the perfect recipe for increased mercury methylation. And that was one of the main studies that Carla Pereira and collaborators published recently. They analyzed the chemistry in the Venice Lagoon, simulating these mosse closing events. They did that by sampling sediment cores from 2 sites. Ahí, pointer. So they sampled 2 sites, Orsellino and San Giuliano. Orsellino is marginal, closer to the coast, and San Giuliano receives more influence of Adriatic seawaters. They sampled 3 cores that were called fresh. They were untreated, they were measured right away, and they sampled 3 other cores that were incubated in oxygen depletion conditions, simulating this mosse enclosure. They were incubated for 48 hours. And from that, they analyzed different chemical aspects related to mercury metabolism. One of the key findings was that they found higher mercury methylation rates in these incubated cores, as you can see in this plot on the right, especially the solid points represent incubated sediments. X-axis represents mercury methylation rates. So especially in 2021, mercury methylation was increased after incubation, and there was more capacity of this methylation in Orsellino than San Giuliano, so closer to the shore. The natural question following that was what's going on at the microbial activity level, and that's what Carla and I are currently analyzing through metagenomes and metatranscriptomes. And the questions that we want to answer relate to the diversity, abundance, and distribution of AGCA gene, which is the responsible one for mercury methylation. We want to see if there are indicators of anaerobic metabolism after these incubations. And let's see if we can correlate gene expression levels of this gene with methylation rates and also other environmental parameters. I'm going to go super briefly over the AGCA part. We've been able to retrieve, I was going to use the pointer. So we've been able to collect 44 AGCA genes from our metagenomes. We've seen more diversity in Ocelino, the one closer to the shore, than San Giuliano, that was the one closer to the Adriatic Sea. But we see higher overall gene abundances in metagenomes in San Giuliano. From all these sequences that we've been able to retrieve, we have built a gene catalog. That means that we have clustered these sequences based on how similar they were at the sequence level. From that, we have found that there is one sequence that is a core AGCA present in all the samples in our study. There are other AGCA sequences that are specific of fresh sediment cores, mixes of samples, but a cool thing that we see is that there's a huge amount of diversity of AGCA sequences that are specific of Ocelino or San Giuliano. By checking the taxonomic annotation of these clusters, gene clusters, we see that this core sequence belongs to the Spirochaeta and the Sulfobacterota, and we also see that there are taxonomic groups that are specific of Ocelino or San Giuliano, like Nitrospirota and Myxococcota. Finally, even though we see this huge diversity, when we check the gene expression of these clusters, we see that actually methylation is dominated by certain gene clusters, and we need to do statistical analysis, but we see that there's actually higher expression of some of these clusters that are specific to Ocelino, as we saw in the methylation rates with all the chemical assessment of the Venice Lagoon.
Okay, so this is where we are actually in the current knowledge of mercury methylators. So sometimes we find them, sometimes we don't find them, sometimes they are abundant, sometimes they are active. Some of them are specific to specific hotspots, and some actually are quite versatile, and we find them in the several ecosystems. But what we know for sure is that while these are the two sites, Ocelino and San Giuliano, when the oxygen decreased during the incubation, methylmercury concentration in both cases increased. Okay, so as most of the ocean, so we have studied the anaerobic environments, what are really present and expanding, and this is very important. But okay, most of the ocean is enriched in oxygen. So what is happening in the global ocean? So we can see here is the oxygen concentrations across the globe, and of course higher concentrations of oxygen are found in cooler waters because the oxygen solubility depends on the temperature. So lower oxygen, but still at 200 micromolar, it's very high for mercury methylators. Okay, so let's see what we know about the Oxy global ocean and what we actually found. The first study that went and searched for the AGCA genes was Podar et al. And they found it in very few sites, actually, in marine water columns only, in 4 sites. One is an oxygen minimum zone, the other one is the sunny inlet. And they actually, believe me, this is the Malaspina, and they found it in very low concentrations. Later, in 2020, actually, thanks to the Tara Ocean efforts, they searched for the AGCA gene. And they found that actually, Marta explained about the cluster. Clusters are kind of microorganisms that they are very similar with this gene. Cluster 3 was the most abundant, widespread across all ocean basins. But what we know about this cluster 3, that most of these microorganisms are Nitrospina, for which they have never shown the capacity to methylate mercury in the laboratory. So then we realized that something was going wrong with the methods that we were using at that time. So we checked what was the protein that's responsible and what they have in common and what they didn't have. And actually we observed by compiling a large data set of all the cultures and marks and metagenomes of microorganisms that were supposed to be methylators, that only true methylators were 32. And in these 32 methylators, this protein had 4 conserved regions. So in order to reach an agreement and to not publish not true mercury methylators, we went to this very restrictive criteria in which they decided that only the microorganisms that were having the gene and these specific sequences would be mercury methylators. What happens then in the oxic global ocean? That we don't know who are methylating because they don't have these regions. But methylmercury is there. You can measure, if you go to the ocean, you are going to find methylmercury concentrations across ocean basins, even in the Arctic, where it's mostly saturated and enriched in oxygen. Methylmercury concentrations change across depths, so for sure there are processes occurring within the water column that we are not understanding. What is happening? How is this gene and how can we find it? So, if the water column is saturated in oxygen, there must be a place in this water column where we should find these microorganisms that do not work with oxygen, but without oxygen. So one study in 2019 just unveiled, say the global niche of marine anaerobic metabolism expanded by particle microenvironments. So if this is a particle, what you can see here, it's a gradient, actually, of oxygen, in another way, electron donors and acceptors. And oxygen is consumed very quickly within the particle, and you cannot measure oxygen concentrations in a particle. What you measure is the decrease in oxygen, here in blue, in green, here in orange, the decrease of nitrates, and of course the decrease of sulfate, with some sulfite production. Me too, I do that. Okay. So can we find mercury methylators in particles of oxic water columns? Okay, not yet in the ocean, but we have found this a couple of times in lakes. This is a work that we did in Lake Geneva, in which we measured mercury methylation rates in sinking particles and in gray, and we compare them with sediments in black. And actually we observed at 3 different points that sinking particles, these settling particles, had a high capacity to transform inorganic mercury into methylmercury. Few years later, then we searched for mercury methylators there, the straight ones, the ones that we know that for sure they methylate. And actually, the good news is that we found mercury methylators also in sinking particles, so what it's called trap, but the abundance did not correspond to the rate. So they were much more abundant in sediments compared to sinking particles, even though sinking particles were a hotspot. But the good news is that we didn't know is that these microorganisms are inhabiting these sinking particles of oxic water columns. So what in the ocean? Okay, this is the Plan Nacional I got recently. It's called PlayMer, and the main aim we have actually is to search for these players and drivers and niches of methylmercury in oxic marine water columns in the context of oxygen depletion. So the hypothesis is that we have this redox gradient, so these little dots that you see here represent a redox gradient of different availability of electron donors and acceptors, and this red is a mercury methylator. The main hypothesis, or the main working frame, is that we have oxic, anoxic, and anoxic and sulfidic waters in which methylmercury concentrations increase, the oxygen decrease. This is the geochemical context. And what about? Okay, this already know. We have preliminary works, previous works that have shown that, of course, they are in both water columns, more abundant in particles. We don't know what is happening in the oxic water column, and the Plan Nacional aims to find those, because so far we didn't find them there. We are going to do this in the Eastern Tropical North Atlantic oxygen minimum zone, especially in this transect in which previous work from Bowman et al. Have shown hotspots for methylmercury. This is the plot for methylmercury concentrations with higher levels at these depths. And if everything works well, Vienna is putting a lot of effort on acquire in situ pumps for the institute, and I hope, and we hope, that we'll have more than one in situ pump to collect the particles and study more in details these particles. The idea is to compile a lot of data set we already have, that we have been studied. You can see Baltic Sea, Kariako Sea. We have 2 campaigns already performed in another Baltic fjords. Mambo, that has been in the East-North Tropical Oxygen Minimum Zone, South, and all these campaigns that are mainly oxic. So we are going to focus a lot on the datasets that are available and on the campaign. So this talk, it was about progress and challenges in understanding methylmercury formation in the ocean that we together have tried to give you an overview of the mercury cycle, to understand the inputs and outputs, highlighting the role of coastal aquifers and the hydrothermal vents as uncharacterized sources of mercury, as the surface microlayer as something that is controlling completely the flux of the basin, that we have worked a lot to really know what it is a true or not true mercury methylators, that we have worked on a consensus protocol with the international community to provide accurate and true mercury methylators, even though we know we might be missing some of them. That actually, in a few of the cases, the mercury availability and the transcript can finally predict the concentration of this toxic contaminant, and that we should not overlook the fact that the global change is affecting the whole mercury biogeochemical cycle, because the oxygen depletion may increase methylmercury concentrations in the whole global ocean. Okay. Challenges, this is a big challenge, is measuring mercury concentrations in the ocean, which are at very low concentrations. There are concentrations of picomolar and femtomolar. This is an exercise that was performed within the GeoTraces program, in which the community has tried to do an intercalibration. So these are the results on the mercury concentrations of different laboratories. A lot of the laboratories agree, but still, you know, this is log scale. So there is one order of magnitude. You might have double, triple, 4 times the concentration in one laboratory concerning to the other. We are working a lot to measure and characterize at these very low levels the concentrations of mercury at this institute, at the ICM. There is a new analytical service that is possible thanks especially Gemma, and then Celine and Julia, who have been working a lot on making the standards, calibrations, checking with samples, and this is also possible thanks to the Geologia, because they host the instruments even though we have very little space. And we don't do it only in the lab. We are able to bring the instruments to the boat. This is the last cruise, Pantok. Gemma showed the data of the last cruise. These samples were measuring on board with Emily. Challenging milieu of the boat is contaminated. You cannot use the milieu of the boat to do your calibration curve. Then the atmosphere is not necessarily clean, so if you bubble a sample, you contaminate your sample. A lot of challenges, but working a lot on this. Gemma with Emily. And last, okay. Mercury is a contaminant. We study mercury in the ocean. How we reach the society? Because so far, I think Jorge Fernandez is reaching the society much better than us, that we are really working and putting a lot of time on knowing what is the impact of this contaminant. So we are trying to. So every time we do on a cruise or we interact with not scientific people, we collect their hair. And we have been doing that in cruises. So I did it in 2 cruises, and Gemma and Emily did it in another cruise. And then what they get, we send them an email with their concentration, of course X, because it's the example. Here in red, we tell them how is his value compared to the Spanish average, which is 190, with the maximum value and with the lowest value. And then we, of course, tell them that it is extremely important to eat fish. Fish is a source of omega, is a source of vitamins, but we tell them which kind of fish they should eat. And we highly encourage to eat fish white and blue, I don't know, sardines, boquerones, and we actually don't tell them to, or we tell them to avoid these fish species from which they can have a higher mercury intake. And with this, I hope you keep the take-home message that mercury is important still nowadays to understand the cycle, to actually control our consumption and continue understanding or trying to understand what drives this methylmercury. Thank you.
Thank you very much, Andrea, for this interesting talk, and also to the Mercurians for the participation. So we have some time for questions.
So thank you, very nice talk. So when you say that some microbes, so it has not been demonstrated their methylating capacity, is because they don't have isolates, or because they have and they have tried and they don't do it?
In the case of the nitrospina, this famous cluster 3, it's isolated, okay. And the isolated strain doesn't have the capacity to methylate mercury. Okay. Maybe there are other strains, but the strain that they have isolated and tested do not methylate.
Okay.
Put maybe a slide on this. This is the bioaccumulation. So methylmercury can get into the plankton just because it can cross the membrane. And then all the organisms that eat plankton. And then all that eat zooplankton eat phytoplankton. And then you have the other, and then you have the other. And what tuna is having sometimes is a fish like this that might already have 2 ppms of mercury. So it's just increasing, it's exponential. But the concentration of tuna is 1 million, 10 million, 100 million higher than you have in the water. But it's stuck in the fatty acid, so it's not away, it doesn't get away.
Is it stuck in the muscle or in the...
In the muscle, but in the liver a lot, because while detoxifying, a lot of the mercury gets into the liver. You can measure it also, of course, in the digestive transact.
Yes. One question. You explained the important role of the sinking particles. Okay. And you explained that in your project now you are going to study these particles with in situ pumps. So no, my point is that in situ pumps sample suspended sediment, suspended matter that is in the water, and that can be in the water for long periods of time, organic gels, or I mean many different kind of substances, but sinking particles And taking sinking particles perhaps is not the best technique to do it. I mean that all the sinking particles that are produced in the water column due to, I don't know, biological aggregation, fecal pellets, by physical, chemical processes, all this can settle quite fast, and you don't take them with the in situ pumps.
Yeah.
So then, have you worked sometime in material collected in sediment traps?
This actually, Albert, this is a very good question because perhaps I didn't explain very well the differences. The work I presented, about sinking particles were really sinking particles, actually. These were really sinking particles because we didn't have in situ pumps in Lake Geneva, so we put the traps. And we let the traps for 28 days. So there we collected exactly what it was sinking, okay? And that gave us the first hint, okay, the rates are very high. First is that the first thing we measured, and then we searched for the gene. Okay, rates are high, but they are this microorganism. In the case of the question of what are the methylators in the global ocean, we are not searching or targeting sinking particles. We are targeting particles. We don't care if they are sinking or not or floating, because we just look at the particles as anaerobic microniches, where these microorganisms can be. And they can sink or they can float and they can stay there and methylate. So exactly, the Plan Nacional just searched for particles without making the difference. That they have a difference because perhaps some of them are more degraded or less degraded, but the idea is just to find the niche where they are, because we don't understand if this is biologically formed, where it is happening in the oxic water column of the ocean. Yeah.
Thank you for the talk. I think it was great. I have a question regarding the balance of mercury in the ocean. You said that the ocean can get mercury through atmospheric deposition, but also the ocean can produce and be a source of mercury. So at the global scale, would you say that this is balanced, or if the ocean is more a source of or a sink, or what are your thoughts about this?
Actually, the efforts to make the global mercury budget have put a lot of weight on the evasion because we don't know the balance. So if we don't know so why we have more mercury is because it's anthropogenic or not, and how much of this mercury is evading. The evasion is not measured. What we said is evasion is what we suppose it should be there, and we don't find. So it's not closed. Yeah. But the ocean is a reservoir of mercury as a whole. And mercury levels in the ocean have increased dramatically in the last 100 years.
I just have another question. I mean, I think that one of the paradoxes that you find is that there is a lot of methylmercury in water, in oxygenated water, and you don't know how it is created. And then you assume that it has to be in particles and micro-oxic niches. Could it be that it is something that is happening at very low rates in just oxygenated water? Is that possible, or?
It hasn't been demonstrated, even though it has been. So we have, not me, but the community has tried to do incubations to see whether methylmercury can come from inorganic mercury from an abiotic reaction, and we don't find it. Perhaps it's so low that we are unable to measure it. That's a possibility. What it has been demonstrated is that before we thought, I didn't talk about this chemical species because we don't work especially on it, and it's quite complex, but dimethylmercury exists. So actually, two molecules of methylmercury bind, and they make a volatile species of mercury. Dimethylmercury is volatile, so we generally think, okay, and then it leaves. But no, it cannot live at 4,000 meters depth, right? So that could be another potential source of this recycling and forming of methylmercury, that in generally is not well studied, neither, because dimethylmercury is very low, 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 and we have hard time to measure it too.
Thank you, very nice talk. I don't know if you have mentioned this, but which is the role of the photochemistry in the mercury cycle?
Yeah, role of photochemistry is important at the surface of the ocean because I'm trying to get one, this one for example, because both reaction, this degradation and this degradation can be mediated by light, but as a whole, most of the ocean is aphotic. So we speculate or we assume that most of the degradation of methylmercury at a global scale will be dominated and driven by the microorganisms that carry the genes responsible for methylmercury degradation. Can I add something to this?
You can.
To Chris' question? Because I prepare it, in case. Because we work on this here, actually. So we studied already the distribution and abundance of the geography of the genes responsible of degradation of methylmercury. And they are highly abundant across all ocean basins. This is the MERV is responsible for the degradation of methylmercury to inorganic. It's abundant and is expressed metagenome versus metatranscriptomes. And same for the MERE. Isa worked on this when she was here, actually.
Is more degraded in the microlayer?
We should measure methylmercury concentrations from the study Gemma is doing. It's the next.
I also want to add something about what Fran asked. That also it's difficult to know these fluxes because mercury is zero. It's difficult to measure because it's really volatile. So maybe this is also a problem why we don't know the amount of mercury that it's released into the atmosphere.
Yeah, it's really little concentration. Gemma tried to measure it, especially in the surface microlayer, but it's so low compared to the total chemical species. And by the fact of bubbling, this is the technique to measure it, we can contaminate the sample, so she could not manage. But keep on trying, yeah.
Okay, thank you very much, Andrea. I think it's time to leave it here. Have a good tuna fish for lunch, and thank you very much for your presentation.